Okay, good morning, everyone. So, shall we start? Okay. So, by this time, I hope you understand the basic concept in tumor immunology. So, today, we are going to discuss immunotherapy, and we cannot do any therapy till we know the biology, and you have to recall everything what you have learned in last 2-3 days, okay? So many of the targets which we will be discussing, they are not going to be new ones. Basically those are targets we already have discussed with you, and hopefully you will understand it better. So when it comes to tumor immunotherapy, what exactly we mean is effective as an adjuvant or part of the conventional therapy. That's the one key element in tumor immunotherapy. Keep in mind that it is not going to be a monotherapy. Okay? It will always be given to patient along with other treatments. And what are the conventional treatments for cancer? Chemotherapy, radiation therapy. You forgot about surgery. And other targeted therapies, okay? Because that has also, now they are becoming more and more, conventional treatments. So tumor immunotherapy facilitates the destruction of hard-to-treat tumor foci, which escapes conventional therapies. And that's actually the beauty of tumor immunotherapy, okay? Because as I have mentioned many times, that our conventional therapeutics, what we have for cancer, will not be able to kill all cancer cells. There will be cancer cells which will escape. So all these patients, they have leftover disease, hard to reach micrometastasis, or minimal residual disease. And only cell types which are capable of finding those cells and trying to destroy them will be your immune cells. If we can arm them correctly, if we can activate them in a right way. The complexity of immune response against tumor necessitates a multilateral approach to solve the problem of immunotherapy. I hope by this time you can understand how complex immune responses are. Okay, so it's not going to be, one approach, one, target which can have all the success. So it has to be finely optimized and balanced. Only then they can be more effective. And at the end of the time, basically what we want to do is that it should enhance its specific and non-specific host response and minimize the tumor escape. Even if it is not capable of killing all the tumor cells, if they can restrict their progression, that's good enough. That can prolong the survival. And actually, all the tumor immunotherapies which we have today, they are not cure. Okay? They are not curing the patient. What they are doing is that they delay, they are enhancing the survival. And enhancement of survival, when you think about it, that tumor is still growing, but just growing at a slower pace. If we can slow down that pace more and more, probably those patients will survive longer and longer. So don't think that even immunotherapy is going to go and kill all cancer cells. It's not possible, because cancer cells are very smart. They will always find some way to escape, some unknown mechanism which we still don't know. They will find it, okay? Because that's what they do. But goal is that if somehow we can, make these people live longer, restrict the progression of the disease, that will be good enough. And if you remember, just recall your whole concept of immune surveillance. Immune system, even if it is not capable of eliminating all the cancer cells, they hold the tumor. That's what causes the latency. Otherwise, by the time transformation happens, everybody will have a full-blown cancer. So this is a cartoon showing different history of actually tumor immunotherapy. The first tumor immunotherapy which was tried was colitoxin, which is just a mixture of bacterial products. And what it was doing is that it was inducing tumor necrosis factor and interleukin-1 production, like creating inflammatory milieu. If we look at broadly here, there are 4 different arms of tumor immunotherapy. One will be your non-specific, in terms that you are non-specifically augmenting immune response by using methods to enhance inflammation, different cytokines which are used in immunotherapy. They will all come into this category of non-specific immunotherapy. They are not specific for a specific antigen. Okay, keep that in mind. Non-specific immunotherapy will be not specific to a specific antigen. Then you have a whole class of passive immunization, which are based on the antibodies, and this arm is actually widely used in many different ways. The antibodies is specific for specific targets on the tumor cells, have been utilized to kill tumor cells, restrict their growth, or deliver different therapies like radiotherapies or toxins to the tumors which are expressing those antigens which can be identified that antibody. There is a whole class of antibodies which in itself are able to inhibit cell growth, and they are widely used in our hematological malignancies. So this is a huge area where there are a lot of antibodies which are in clinics right now. There is another branch where there is active immunotherapy, which is vaccine-based. And when we talk about vaccines, tumor vaccines, what actually we are looking into is that there are 2 ways you can utilize vaccine approach in tumor, in managing tumors. But in majority of the cases, it will be therapeutic in nature. So basically, you are developing or designing vaccines which are going to cure or at least control tumors in patients who already have it. So that's called therapeutic. In reality, most often when you think about vaccine, you are thinking about prophylaxis. You get vaccinated so that you don't get that disease. That doesn't happen in cancer. There are some prophylactic vaccines which are indirectly or directly, limiting, tumor. And do you know an example of any prophylactic vaccine? We have discussed it many times actually in this class. Which one? Somebody said the right word, but cervical cancer, HPV vaccination basically will be considered as prophylactic vaccine. Those who are getting also for like hepatitis vaccines, they can also be considered as EBV vaccines, not so much in India, but in African countries, EBV vaccination is still applied. So sometimes, these kind of, if we know the agent which causes cancer, and if you can vaccinate against those agents, probability is that you can prevent that cancer to happen, and that's a prophylactic. And then there is a whole area of adaptive immunotherapy. Basically, in this, you're looking into, taking the cells, immune cells like T cells or dendritic cells, manipulating them and transferring them back into the patient. So like a transplant, could be autologous, could be allogenic in nature, and This, you can do it ex vivo manipulation in many ways. You can simply, activate them, like you saw what they were doing early on with something called lymphokine-activated killer cells. Basically, they were isolating lymphocytes, activating them with IL-2, and then infusing them back in those patients. Or you can take, T cells and manipulate the receptor so that they can recognize only antigens which are expressed on the tumor cells. And you can do that especially for those tumors which are monoclonal in origin. Like most of them are expressing exactly the antigen which can be recognized by that, that engineered T cells where you have manipulated T cell receptor. And that has been utilized. And also, the CAR T cells will come into this picture, which is, the hot area right now. All those immune checkpoint blockers will come into this or this category. Which category they will come? This is your passive immunization. This is your active immunization. Your immune checkpoint blockers, what do they do? What's the role of those immune checkpoint blockers? What do they target? Co-stimulatory molecules. You remember? And one of the co-stimulatory molecules which is immunosuppressive is your CTLA4, PD1, PDL1. In those, they will come into in this category where passively, they are trying to enhance the immune response. There is nothing antigen specific in that way. So this is the basic, tree which you see, what we have today, what is started very early on in 18th century. And still this area is growing, and a lot of success has been achieved, at least in last, I will say, 10 years. A lot has been done, and I will show you some of the examples tomorrow. What are those? At least 10 or 15 different drugs which have already been clinic, making lots of money. So let's recapitulate what we just talked about. When we talk about tumor vaccine, most often we are talking about it as a therapeutic vaccine. This has been tried for long time. Early on they were using just inactivated tumor cell, like what you do, when you are trying to generate, antibody. And most of the vaccines which we get, for all the other diseases, those are all inactivated pathogens, in many ways, which are mixed with adjuvants and given to us. They tried to do the same thing with tumor cells, and it works. At least in a preclinical model. In mice it works, but it doesn't work in human. Designing means to make tumor cells more immunogenic. And there are ways, I hope you can imagine ways how you can make a tumor more immunogenic. Just try to block or fix all those factors or characteristics which causes these tumor cells to become non-immunogenic or escape. I gave you the whole list of things, in your, I think, second lecture itself. But more important, I think one molecule which have been exploited to make these tumors more immunogenic is MHC class I expression, because in most cases the tumor cells, they downregulate MHC class I, which is necessary for antigen presentation, and only then these tumors can be recognized. Otherwise they will be blinded. Immunization with defined peptide in a novel adjuvant to generate cytotoxic T cells, and this is a very open-ended. People have tried to use specific peptides combining with different adjuvants, and they have been successful in generating T cells, and some of those vaccines are in clinic and also in clinical trial. But the key point in all these aspects, whenever they are trying to design these tumor vaccine, is to find a way to recruit, right type of immune cells to the site of tumor or metastasis, do the right expansion and targeting, and they start with antigen-presenting cells, T cells, and all the other cells which have to be there. But at the end of the day, their goal is to generate these cytotoxic T cells, because these are the cells which are capable of going and killing the tumor cells. The prophylactic vaccines which have been used to prevent cancer formation, most of them are against those viruses which have been linked to different type of cancer, like hepatitis B for hepatoma, papillomaviruses for cervical cancer, and other targets actually include your Epstein-Barr virus, HIV, or HTLV-1, because there are many malignancies which are associated with HIV or HTLV, and EBV is associated with B cell, a specific type of B cell lymphoma. So here is a list of non-specific ways how you can stimulate immune response, and this is a short list. List is pretty long, but I hope it will give you some idea, what are the different approaches which have been used. Right in 18th century, as I was said, that colitoxin was nothing but a primitive form of BCG, which have been used because all it does is that it induces inflammatory response, will generate a lot of different inflammatory cytokines production, which have been shown to directly kill the tumor cells or modulate the tumor microenvironment. Different adjuvants have been explored, right from the Freud's adjuvant, detox, this QS21 is in a clinical trial, and LM is widely used. Cytokines have been used, and the most common ones are your GM-CSF and G-CSF. All those patients who undergo chemotherapy or radiotherapy, the first toxicity they will ever notice, of course, you can see they lose appetite, they don't feel hungry, they start to lose hair, right, hair loss. But what happens to their bone marrow? So if a patient who is getting chemotherapy or radiotherapy, what will happen to their bone marrow? Bone marrow will be also affected. And you all know that all immune cells, majority of them are derived from hematopoietic stem cell, which arise in bone marrow, right? So there is a word used as all these patients who are undergoing chemotherapy or radiation therapy, they have a common phenomenon called pancytopenia. What does it mean? Pancytopenia. Have you heard about this word? No? So pan means broad, right? Pan, as whenever you use word pan, it means broad. Cytopenia. Cyto is cells. Penia is what? Loss or low number. So overall you will see low number of white blood cells. And why do you see low number of white blood cells? Because bone marrow has been compromised. So by definition, all chemotherapies and radiation therapies are myeloablative. Okay? Because they will, try not only killing dividing cells, which are cancer cells, but all dividing cells, which will include cells in your bone marrow. Okay? And the first population which gets affected will be which cell type? If a hematopoietic stem cell is killed, which cell type, or not killed, but at least, their cell division is blocked, which cell type, which immune cell type will be affected first in your circulation? Which one? B cells? T cells? Okay, so this side wins and this side loses, okay? Do you remember half-life of neutrophils? How many neutrophils are produced every day? Billions of them. And if it means they need to be produced every day. So in pancytopenia, if you say neutropenia is another word, that these patients usually will have neutropenia. And there are two commonly used agents, which whenever they see this patient is having neutropenia, these patients, they get these GM-CSF and G-CSF, okay? Either granulocyte colony stimulating factor or granulocyte macrophage colony stimulating factor. And what it does is that it mobilizes and maintains, those hematopoietic cells, generates new neutrophils, and then that person, all of a sudden becomes better actually. So this is used as a support, palliative care for those patients who are undergoing chemotherapy and radiation therapy. And so it's a big market. There are a lot of activity in this factor. Another one which is commonly used in these patients are called erythropoietin, which is important for red blood cell maintenance. Cytokines have also been used. IL-2, I have discussed about it, what IL-2 does. It's important for T cell proliferation and immunity. Interferons are also in this class. Different fusion proteins have been generated and used. Anti-tumor and monoclonal antibody to IL-2 is best example. Chemokines have been also exploited, and common one is MIP-1 alpha, which is CCL2. Many danger signals. Again, what these danger signals will be doing is that trying to induce inflammatory response to make these tumors more hot. CpG motifs basically is important because it works through those TLR9 receptors. Co-stimulatory molecules, trying to express these co-stimulatory molecules directly by transfecting tumor cells or infecting tumor cells, or finding a ways to upregulate their expression, because that loss of co-stimulatory molecule is one of the phenomenon which tumors use to escape. Immune checkpoint blockade, we will discuss more in detail, but the first immune checkpoint blockade antibody which was developed was against CTLA-4. And that CTLA-4, when it binds to what? What it binds to? CTLA-4 binds to what? B7.1 or B7.2, okay? Either one of them will be fine. Basically, that prevents, and this was developed in early 2000 actually, and that actually was the basis for what Nobel Prize was given last year. And actually, we share some of the things with this. The person who was also part of that team also did PhD from the same lab where we did. His name was Alaparthi Suresh. Suresh Alaparthi. This was developed by Bristol Myers Squibb, and he still works in Bristol Myers Squibb, and he was part of that team. Usually it's not one-man show, when you are working in these kind of big projects. So he was part of that team. Cyclophosphamide, which is a chemotherapy drug, which have been used, BCG, liposomes, and FLT3 ligand. Some of these red ones which you see, I have been part of that, some of those preclinical studies, before it went to the clinical trials. So that's why I marked them red. And I know a little more about them, how they work and what exactly we got. So if you have any question, we can talk about it later. When we talk about the passive immunotherapies, there are many, many different antibodies which are currently used. Here is just a sample of it. In most of these cases, these antibodies are often tagged with either drug, or in itself they can inhibit. They have been used as a neutralizing antibody. And here is some of those billion-dollar. Each one of them has a market of more than $1 billion. Herceptin, Rituxan, Erbitux, Compath, Javelin, Bexar, myotarg. And actually, you can see many of them are for hematological malignancies. And one of the benefit is that most of those tumors, most of them are B-cell, malignancies. And most of the B cells, they express CD20, so it's very easy to target, and it is a neutralizing antibody. Herceptin, almost 25% of all breast cancers, are positive for Herceptin, and it is widely used in managing, aggressive breast cancer. EGFR is a growth factor receptor, which is overexpressed in many solid tumors and has been widely used, and it is approved. CD52 and CD33 is also a specific group of leukemia and lymphoma which have been targeted using this. There are other antibodies which are also targeting the different steps in tumor progression. Avastin was a multi-billion-dollar drug when it came. This was one of those anti-angiogenic antibody against VEGF alpha, VEGF-A. And it was used with every solid tumors and also in other malignancies. It has gone out of favor a little bit, at least in cancer therapy, because there is not much survival benefit which have been seen by using Avastin. But it doesn't mean that this antibody is still not making money. Think about there are other diseases where abnormal neovascularization plays a role. And so right now, the only treatment for glaucoma, eye disease, is this antibody. So this antibody now is very much used in ophthalmology. And that can be so it all depends on which disease. So early on, they were developed for cancer, but not they're using for Avastin. There are limitations for this therapy, and the most important is, these antibodies are large molecules. The delivery is a problem. Getting to the site of tumor is often very poor, and some tumors are very hard to reach. And your best example will be if you look at pancreatic cancer. Okay, pancreatic cancer is the best example where nothing reaches actually to the cancer cells, because when you look at the tumor, only 10 to 20% of the tumor mass are the tumor cells. Rest of them all are like a fibroblast. It's a very solid, hard block of cells, and most of them are fibroblasts. But that cancer kills, basically within 6 months. So it's that cancer, there are certain cancer types and many solid tumors in particular. These antibodies have poor penetration. They can bind to other cell types. That's the reality, because that antigen can be expressed on other cells, and then these antibodies can go and bind, and that leads to toxicity. The best example will be this EGFR. It's not just expressed on the tumor cell itself, but will be expressed on other normal cells also. CD20, of course, it is a B-cell, marker, so it's expressed on B-cell lymphomas, those tumors, but also will be expressed on normal B cells. So you are basically looking into those patients who are getting treatment with these antibodies will have B cell aplasia. Basically B cells will be lost in these patients. But don't worry, okay? There is a nice treatment for B cell. Anytime you lose B cells, they can find a way to supplement it, okay? And it's commonly used. Do you know what it will be? What B cells produce? Antibody, okay? Is it possible to get antibody cocktail, which is clinically used today even? Yes. So basically, they have a cocktail. They get it from actually AB positive individuals, donors. Their serum basically is acceptable to most. Don't ask me how it works, okay? But it takes care of these individuals who have lost B cell population, and usually these kind of loss, once you withdraw these treatments, B cell population will come back, okay, because your bone marrow is not compromised. It's only those mature B cells, which are having these issues. And antibodies are often immunogenic, So there is a antibody to antibody, anti-antibody, generation, which happens very often. It's a very common phenomenon in mice. In human, it has been fixed actually now. So basically, they don't use anything which can generate, antibody to that particular antibody. They use now these antibodies which are commercially used. These are all humanized. They call it humanized monoclonal antibody. And they do this humanization by, recombinant DNA technology. And they are not using the whole, antibody. It's a large molecule. They are just using a single chain FCFB, portion, variable region only, to get the response. So they are not generating that kind of immunogenicity. And actually, that technology, which was used to create these SCFB fragments, is multi-billion-dollar technology, because that technology was patented. This was developed in Genentech. Okay, this is a nice story. So anytime you hear the basic research and how it gets funded. So this technology was developed in Genentech by a postdoctoral fellow from City of Hope. City of Hope is a hospital in LA. So both, City of Hope and Genentech, they own the patent. So anytime anybody is making these humanized antibody, they will have to pay them. Okay? So only 25% of the share of this royalty goes to City of Hope. And that royalty is close to $200 million per year. Think about it, how much money they make with these humanized antibody. Okay? So just technology royalty is close to $500 million. They must be making more than $50 billion market, okay? Because usually royalties is somewhere between 1 to 10%. You don't get more than that. So sometimes what you do and develop in technology can be patented, and if you get lucky, probably you will get something out of it. You may not get anything, because Pune University will own the patent, and you will not get any royalty. But in our case, we do get, okay? I think in US still they promote those individual investigators, so we get little, amount back sometimes. Okay, let's move on. So here is a very simple cartoon showing, tumor-specific monoclonal antibody therapies, and 3 different examples. This is just tumor-specific antibodies. They will recognize the antigen on the tumor cell. Once this complex antigen-antibody complex is done, then all those Fc receptor expressing cells, like NK cells, will come and bind to this complex, and they will kill these tumor cells which have this specific antigen by a very defined process. It's called antibody-dependent cellular cytotoxicity, or ADCC. These antibodies can be conjugated to toxin or chemotherapy, and these can be delivered to the tumors which are expressing these unique antigens, which are recognized by these antibodies. And then basically you are trying to concentrate that toxin or that chemotherapy to the site of the tumor, which will lead to more enhanced killing. And in third way, basically using a radioisotope which have been tagged to these antibodies, and these antibodies will go and bind to that tumor and kill that tumor cells and neighboring tumor cells, because radiation kills cells. So in many ways, in these cases, what these antibodies they are doing is that they are not just targeting the tumor cells which are having these unique antigen, but also limiting the toxicity. Now basically, rather than giving a very high dose of chemotherapy or toxin, you will have to give the optimal dose, and they will not be free toxin or free radionucleotide or just generalized radiation-based toxicity. The tumor antigens which have been targeted by this approach, there is a short list, and list is actually growing. A lot of them are in hematological malignancies, CD5, idiotype, CD52. CD20 is the most common target. Some of these antibodies actually are available here also, have been used in lymphoma management. For solid tumors, it has been always a challenge, and this kind of therapy has not been that successful. except for EGF receptor. This is the only money-making monoclonal antibody so far we have right now. But it has been targeted for CEA or CA125. This is another biomarker very commonly used in ovarian cancer or gallbladder cancer and many other epithelial tumors. Growth factors, the only one which is making money, as I said, is EGF receptor. And there are many stromal components which have also been targeted preclinically, but they have not made it to the clinical reality. So at least in hematological malignancies, this approach has been very successful. In solid tumors, the most successful will be our EGFR and also HER2. Herceptin is also a very popular target and which is making lots of money. So another approach which have been utilized to engineer these antibodies to not just only target the tumor cells, but also to recruit T cells to the site of the tumor. And these are done by using these so-called bispecific antibodies. And these bispecific antibodies basically are engineered where one arm will have the variable chain to recognize the antigen on the tumor cells, and the other arm will have a general activating signal which will cause the engagement of, like here, the CD3. So once you engage CD3, what you are doing is you are activating these T cells, not 4 or 8, okay, all T cells within that tumor, and it will cause that those T cells basically will try to take care of that tumor. And also, like everything else, these antibodies are bifunctional, so they have Fc receptor, and these Fc receptor, once they are engaged, they can bring these macrophage and NK cell or dendritic cells, or all neutrophils also, the cells which are expressing Fc receptor gamma. So there are a lot of activities trying to generate these bispecific antibodies. For most part, they have been only in academic in nature. So there are very few clinical trials which are going on, but theoretically it is possible to design such kind of antibodies and hope that maybe they will be successful. Transfecting the tumor cells have been utilized, and it is very difficult to infect tumor cells in vivo, but at least in animal models it has been tried, to try to transfect them with either co-stimulatory molecules or the cytokines or growth factors which can function as an adjuvant. And the most common co-stimulatory molecules which have been tried is your B7, B7.1 and 7.2. And here idea is that if you enhance the expression of B7.1 and B7.2, probably you will have a better antigen presentation and killing of the tumor. And same thing with the GM-CSF or these kind of adjuvant, is that if you in this tumor milieu or tumor microenvironment, if you have overexpression of these factors, it will recruit better antigen presenting cells and dendritic cells, and they will pick up antigen and do a better job. So that these are basically more or less in a non-specific enhancing the immune responses. Here are the different vaccines which have been tried against unique antigens in combination with the different cytokines, different approaches. For B-cell lymphoma, idiotype with KLH and GM-CSF have been designed. Autologous irradiated tumor cells along with BCG have been given for colon cancer. Intact autologous heptanated irradiated tumor cells have been given in case of melanoma. All they are trying to do is to make this cocktail more immunogenic by using unique antigens or a broader antigens, combining it with the other factors and trying to target and hope that that tumor, that works. There are some shared tumor antigen approaches which have been used, like using the allogenic irradiated tumor cell lines, differentiated antigens in melanoma, and different cancer or testis-associated antigens. All these approaches actually have not made it to the clinic, but it is always good to know what is going on preclinically also, because one day maybe one of them will get the highlight and be more successful. So now this is a general overview about the state of tumor immunotherapy. And now I will gear up and give you some of the modern approaches which is going on right now. And one of them is trying to do this personalized vaccine. And I think we discussed yesterday, as we were in our break, how we can do it, how we can create these. And I was giving example of the Star Wars analogy, okay? So it is possible that you can compare the tumor and healthy tissue from the same patient. You can create, a profile and identify the specific or unique epitopes which can be used to develop a specific vaccine. That's the key take-home point here, okay? There are a lot of words here because there are a lot of technologies which have been involved, okay? And many of them, as of today, are experimental in origin. They are not in clinic, but these are all what future looks like. You are all familiar with next-gen sequencing, right? You have heard about it. It's becoming cheaper and cheaper. So it is now possible to take patient tumor biopsy and healthy tissue and run them through this next-gen sequencing. And what you will get is that you will get all the expression profile, of all the genes which you can ever think about it. And then you can go and compare the sequences which are obtained from the tumor or normal DNA. And then you can identify these tumor-specific, non-synonymous, single nucleotide variations or short intels of protein coding genes. And what it is basically will be that these unique tumor-specific sequences probably some of them will end up being a unique antigen for that particular patient, okay? This is very patient-oriented, and you are doing it for every patient. So you can take these sequences and then you can run it through these HLA binding, trying to identify, which one will bind to MSC class 1 or which one will bind to MSC class 2. And then you can create, or prioritize the potential vaccine target. And this is not going to be one. You are looking into 100 to 1,000, okay? And based on the technology which they have today, designing and synthesizing amino acid sequences are not a major concern, okay? You can do that, and they can do it in a very cheap way. And then you can take these, you can select these and design these unique neo-epitope vaccines. They call it neo-epitope vaccines. Manufacture them in a GMP facility because it has to go in patients, so it has to be done in a GMP, good manufacturing practice, what GMP just stands for, and GLP is a good laboratory practice, okay? So many of these preclinical studies will be done in GLP, but when it has to go to human, it has to be done in GMP facility. And there will be companies, there are some companies actually which are generating and doing and creating these kind of unique neo-epitope vaccines. And this can be done, like the time the patient goes under surgery, gets the tumor out, within a week, they will have all these things done, okay? They will be manufactured and ready to be vaccinated as soon as the patient recovers from the surgery, before they can get adjuvant chemotherapy, or along with the adjuvant chemotherapy or adjuvant radiation therapy, they will also get these neo-epitope, vaccination. And hope that this will not have toxicity issue. That's hope, okay? And that will augment the adjuvant response, response to those adjuvant chemotherapy and adjuvant radiation therapy. Even with all these development, they are not going to be monotherapy even after surgery. They will have to be used along with other conventional treatments because they don't have that kind of faith, clinicians at least. So here is a very simple cartoon. At the time of diagnosis, the tumors as well as the normal cells will be compared, and unique sequences will be identified, and these new epitope vaccines will be generated in GMP facility and infused along with the adjuvant chemotherapy, which is common practice in cancer management today. And another unique thing which there is a high expectation here is that these new epitope vaccines will promote the functional cancer immunity cycle. And this is an interesting concept, is that what it will do is that since it is going to cover all possible, epitopes which these cancer cells have, unique epitopes, which are capable of generating immune response. Hope is that once you infuse these vaccines, they will activate those cells. These T cells will traffic, will go to the site of the infection, will kill the tumor cells, and the tumor cells will release again antigens. And again, since they already have these cells which have already been trained by this vaccination, there will be memory to it. So as soon as they will see this, release of new antigens, they don't have to go through the whole boot camp again. Those memory T cells will, get re-energized and will enhance or amplify the whole immune response. So this will be like a cancer immunity cycle. That's what they are talking about. So idea is that, these, novel vaccines, personalized vaccines, not only will target, the tumors which so far not been able to be targeted, but also will enhance overall immune response against that tumor, and the outcome could be far better than they expect, we can presume, and it will be synergistic in nature. So the goal basically is to ensure a self-propagating revolution of dysregulated cancer immunity cycle. There are a lot of big words in this, okay? But when you are trying to do something new, you will have to use these catchy phrases, okay? Only then you get attention, because it's all market driven, okay? We cannot just do it by just doing research in the lab. This has to be sold to big pharmaceutical companies or big institutes who have a lot of money, who can support to do all these kind of trials. So that's why these big words have been used in this proposal. This is again a proposal which is ongoing, okay? And it's not going on in our place, but this is going on at least 7 cancer centers in the United States. Right now they are doing it. And since it is so expensive, once those centers will have some outcomes, then it can be used, or it can be done at other centers. So these vaccine-induced neoepitope, they will basically idea is they will have CD4 positive T helper cells, and that can basically will limit or can enhance or potentiate overall vaccine outcome. Another aspect here is that important one, what you have to remember is that by concomitantly, like by simultaneously inducing CD4 and CD8 and CD4 positive T cell responses, these vaccines may contribute to tipping the balance from tolerance to productive immunity. So when they are trying to take those epitopes, they are not just restricting it to MSC class 1, but they are also including MSC class 2, So MSC class 1 basically will, potentiate the CD8 positive T cells, but MSC class 2 will potentiate your CD4 positive or CD4 positive help. Because this is long, usually CD8 positive T cell responses are short-lived, okay? CD4 positive T cell responses are long-lived. That's another important point. So idea here is that by activating or inducing both of these T cell responses, they can have a better outcome and better survival. If we look at some of other big expectations here, is that conventional stratified medicine matches a patient to existing off-the-shelf drug using biomarker assays. And this is what we have right now. Most of the vaccines, tumor vaccines which have been tried right now, they have been developed against certain specific antigen that has been used for a broader group of patients. If they have that particular type of cancer, they get that vaccine. And that's the approach they have. What this personalized one is trying to do is trying to create a new form of vaccine based on these. They call the word they use is mutanome vaccination, neoepitope vaccination, mutanome vaccination, which will be a patient-specific therapy that can target cancer mutations per se, irrespective of their primary sequences. And this can be personalized to the specific patient, not generalized, to a broader population of patients who are having, tumor cells positive for a specific biomarker. Thus, these vaccines may qualify as a universal or tailored therapy from which each cancer patient may benefit. Again, this is a Star Wars idea, okay? But trust me, it's a doable, and it is all possible because of the fact that technology is changing, okay? 2 years ago, nobody thought that next-gen sequencing will become so cheap, okay? Today, it's a reality. It is possible to get these done cheap. I don't know how much it costs here, but in our place, it's not very expensive. It can be done for each patient, and generating those, peptide sequences, that's not also expensive. Identifying those sequences are the difficult part, and that's where actually a lot of help will be needed, because whatever algorithm which have been used are what we have today. It may not be correct one, but it is also evolving. That's another area, which broadly called bioinformatics. There are a lot of people who are working behind it. Only good thing is that there are a lot of people who are working because there are a lot of interest. Bad thing is that most of those people who are working on those projects, they have no idea about immunology or cancer or even biology, okay? 99.9% of them are computer science people, okay? For them, everything is association, correlation, and all those distance. They are very much defined by patterns. They are just trying to establish specific patterns. So they are very far from biology. So hopefully some of our biologists will end up being a good bioinformaticist and will have a better outcome. So this is actually the one which we were just discussed. Today what we have is that we pick up a specific marker, develop a vaccine, and give it to the patient, even if they are negative, positive, or in between. Personalized one basically will be very unique for each one of these patients, and hopefully the outcome will be very different. And actually everything is going personalized, these days, and it's a big catchy word. One of the things is that there is an interconnectedness which is linked with our cancer heterogeneity. We all talk about cancer is a very heterogeneous in terms of not just the cellular phenotype but also in responses. So there is a possibility that that cancer may one respond to this treatment but will not respond to the other treatment, and maybe these kind of approaches are helpful but can also pose challenges. One of the things here they are discussing is that the complex interplay of these sources of interpatient heterogeneity affects both course of the disease and efficacy of immunotherapy and calls for personalized approaches. And the best I can show you in this cartoon is that if you look at the tumor heterogeneity, nothing is clonal, at least in solid tumors. So every tumor cell is very different. It creates a very immunosuppressive environment. And if you create a curve between all the different somatic mutations and all the hereditary or environmental factors, you are basically creating a huge diversity, a very heterotypic nature of the different patient population. So if you combine all the different factors which play out and give them proportionate response, basically you are looking into not just each patient as a different patient, but within the tumor also there are a lot of heterogeneity. And I can tell you that if they are really successful in creating these kind of neoepitope or these unique vaccines, that will be able to take care of this heterogeneity at an individual patient and at the tumor level. So that's the future, at least in tumor vaccine, which we are talking about. Right now, most of the therapeutic vaccines which were approved, actually there was only one which was approved, Spirocell T for prostate cancer, and that company also closed. So there is not so much going on in trying to have a tumor vaccine which will be therapeutic. So that's why these future approaches are more promising, and hope is that if they can make it more individualized and more personalized, outcome will be different, because so far outcome has not been very good by using tumor vaccine as therapeutics. There are a lot of success stories, but most of those success stories in immunology are by blocking immunosuppression, trying to enhance immune response, by preventing cancer development, but not once you have the cancer, just directly targeting in an antigen-specific manner, like creating tumor vaccines. It has not been very successful. That's why these high-tech approaches are more promising. Okay, so we will take a break, and then when we come back, we will talk more about adaptive T-cell transfer approaches. And in all these immunotherapies, there is a side effect associated with it, and we will have some discussion on that, because anytime you interfere with the immune response, you are creating a whole different, pathological condition. Okay. Okay, shall we start? Okay. So now we are going to have some discussion on adaptive T cell transfer, and we will end up with immune checkpoint blockade and talk about some of the toxicity issue in this session. One of the cell type which have been widely used nowadays and very hot right now is called CAR T cells. We will discuss how do they make these CAR T cells in immunotherapy for human cancer. Adaptive T cell transfer is not something new. It has been tried since the cancer actually was discovered or very early on in 2000, I think 2050, 52 people have tried to do some of the adaptive T cell transfer experiments. And basically what they are trying to do is that they are trying to isolate, the T cells from the patient, ex vivo, treat them, expand them or engineer them and give them back to the patient and hope that those T cells will respond better and have a better outcome. As far as the CAR T cells are concerned, the most prominent one or the first one which got approval from FDA was against B cell lymphoma, which targets CD19 on the B cells. And this has been really successful in refractory pre-B cell acute lymphoblastic leukemia or diffuse large B cell lymphoma. There are different forms of adaptive T cell transfer. Very early on when they were trying to do, they were just trying to do these tumor infiltrating lymphocytes. They will take out fresh tumor, isolate tumor infiltrating lymphocytes, expand them in vitro by using interleukin 2 as one of the most common cytokine, and then put them back into the patient and hope that tumor infiltrating lymphocyte activated ones, they will go and have an effective immune response. T cell receptor engineering on T cell has been tried. Basically, they are trying to take these T cell receptors, engineer them to identify unique antigen on tumor cells, and also tag it with the signaling moiety in T cell receptor complex, which is zeta chain, okay? Because that's the one peptide which has the signaling downstream. Rest of them are just like a co-receptor. And of course, these CAR T cells, which is basically chimeric protein, which is created by doing this cellular engineering, which we will discuss. So tumor infiltrating lymphocytes have been shown to induce durable complete response in patients with melanoma. And this trial actually was done in 1970s and 80s, in NIH, and have also been tried even today. Also, they use some of this technology to, get a good response in melanoma. Melanoma is a very good target for many immunotherapies because melanomas are usually more antigenic compared to other solid tumors. The first T cell receptor T cell cancer immunotherapy used in clinic was against, again, metastatic melanoma. Basically, they tried to use T cell receptor that bound to human HLA-2 restricted peptide, and this peptide was isolated from MAZE-3, which is a melanocytic differentiation agent, antigen, as MAZE-3. And this was in HLA-2 restricted fashion. Again, there are a lot of HLA restriction in our patient population, you can see. But this is the most common type of HLA, you can find at least in Caucasian population. 40% of them have HLA-A2. So basically, it increases the patient population where it can be tried. Because you can think about it, each one of us has a different HLA, HLA major or HLA minor. So that also makes a very, these things more restrictive. Definitely, it came with the cost and also the targeting normal melanocytes in skin, eye, and cochlea. And the way they discovered that this treatment works, that many of these patients have white spots. That was like within a week they will see white spots. It means that tumor infiltrating lymphocytes are not just only killing the tumor cells, but also killing melanocytes, which are having the same antigen which they targeted actually. So that's a toxicity. But for the melanoma patient who have a high, metastatic melanoma, basically for them, this is the best medicine they have because survival is very poor. There is no other treatment available. Chimeric antigen receptor, CAR T cells, this basically combines antigen binding domain. You can remember antigen binding domain in your T cell receptor or also in your antibody. Think about that. Most commonly a single chain variable fragment, also called SCFB. This is basically they are synthesizing that portion of your immunoglobulin gene or your T cell receptor or B cell receptor, and they are utilizing that and combining it with the signal domain of T cell receptor, zeta chain, which downstream that will be the only portion which will signal downstream, and also co-stimulatory molecule. So basically, it's trying to create a cocktail, of all the required component, which is essential to, to induce an effective immune response. Okay, so you have antigen recognition element here. You will have a downstream signaling element here, and you will also have the second signal. Think about it, right? So everything is there in one cell. They can create this. And this is all done through recombinant DNA technology. You can clone it. You can infect the T cells. You cannot transfect them. They are always using the lentiviruses to infect these T cells. And this type of synthetic receptor molecule enabled MHC independent target recognition. Again, this basically broadens the patient population and also enhances the overall response. The independence of CAR recognition from MHC restriction endows the CAR T cells with the fundamental anti-tumor advantage, because a major mechanism of immune evasion is loss of MHC molecule. Most of these tumors, they don't have MHC molecule, and if you can remove that restriction, then basically you are making these tumor cells, whether they have MHC or not, a better target for these CAR T cells, and that's actually what happens. Here is the simpler cartoon. The T cell receptor, what I just discussed before when they were engineering, basically they were engineering this T cell receptor epitope, combining it with the zeta chain. But your CAR T cells basically is using these are first generation CAR T cells where you have SCFB having all these antigen recognition domain combined with the zeta chain. They did not have co-stimulatory molecules here. It was not very successful. So they went on to tag those co-stimulatory molecules. Here they did CD28. And also they co-infected the cells with this secondary co-stimulatory molecule like 4-1BB. And if you remember it, the first signal, second signal, both signals are engaged. There is another secondary element comes into picture, is this 4BB, which binds to 4-1BB ligand and 100 times potentiate that whole response. If you go back and look at your slides from your note from yesterday. So this was the second generation, and in this they were doing these separate infections using two different vectors. And in third generation they combined all of these three together. Basically they have antigen recognition epitope, they have the co-stimulatory molecule, they have the downstream signaling, and also they have the secondary co-stimulatory element as a 4-1BB. There are a lot of cellular engineering going on here. These are all done mostly by big pharma industry, pharmaceutical companies. They have capability to do all these. What happens is that when patients come to clinic, and if they become subject for this kind of treatment, clinicians decide that they want to do these CAR T cell therapy. They will isolate, they will do apheresis. Basically they will isolate their peripheral blood mononuclear cells. They will send it to the company, and company will basically isolate the T cell by using fractionation, infect these constructs to those T cells, grow them enough in number, and send it back to the clinic. And clinicians will infuse them in patient. You understand the flow chart, basically? So that's how they do it. It's not done in any academic center. It's not done in our lab, or it's not done everywhere. It's done by, right now there are 3 companies in the United States who have FDA approvals. They are doing it. And I will tell you other costs and other issues which are associated with it. But there are a lot of toxicity involved in it, but those patients who survive toxicity, cure rate is very high. So it has been very successful, and at least it has been very successful in B-cell malignancies and leukemia, because the target is very much, if you know the target, which can be recognized by this, epitope, then you can imagine the response will be far better. So B-cell malignancies as unexpected success. The second generation CAR T cells targeting CD19 has been very successful in B-cell lymphomas. Several features makes CD19 as a very ideal target, because all B cells express CD19. But again, it's not restricted to just B-cell lymphomas. It is also required by normal B cells. So you can think about it also wipes out B cell, which they have to take care of. That's why here I said patients successfully treated with CD19 CAR often have profound B cell aplasia, and they need those gamma globulin injection at that time. There are some variations where most of these clinical trials are going on. I can show you, and it's very interesting, fascinating to know. If you look all the clinical trials in cancer, this is a chart here, okay? All clinical trials worldwide, and most of them are done in North America, of course, Europe, and China is number 3, and India is somewhere here, but still it is in top 20. Which is, so it's not bad. There are a lot of trial going on, but you think about the patient population. Probably India has one of the major patient population as far as cancer is concerned. And clinical trials are important in many ways. They not only bring the new, therapies to clinic, but also creates an environment where you can go and do more discoveries. Because trials are important to find out the errors. Okay. If you don't do the trial, you will never find out what are the errors, what are the problems, and I think that's a very important component. But if you look at the CAR T-cell trial, number 1 is China. There are a lot of clinical trials which are going on in China, and then you have in North America. India, I don't know if there are any centers which are currently doing CAR T-cells, but I'm sure there are a lot of discussion and talk going on, because there are a lot of behind the scene things which has to be developed in order to get these to the clinic, and isolating those T cells, inducing them or infecting them with viruses and expressing them, and again expanding them and sending them back. It's not an easy task. It requires a lot of work behind the scene, and so I think someday probably it is possible. China, it is easier to do many things in China. Sometimes dictatorship is good thing, and democracy is bad, because over there, if one person makes the decision is made, okay, and it can be done. So I think it's easier to get things approved in China. If you can convince a group of individuals. In India, you have to convince armed public. It's very difficult. There are a lot of committees you have to go through. In America, they have same thing. There are a lot of committees we have to go through. There are a lot of toxicity, and you have to justify and before you get to. But there are a lot of clinical trials going on in America and Europe also with the CAR T cells, and so there are a lot of hope in this area. The other targets for CAR T cells, CD22 is actually in clinic right now for multiple myeloma and acute lymphoblastic leukemia. For using HER2 as a target in breast cancer, because as I said, 25% of breast cancer patients are HER2 positive, and it is a good target for breast cancer. And also some of the other cancer like renal cell carcinoma, there is a carbonic anhydrase as an antigen, or in glioblastoma IL-13 receptor alpha 2. But these are still not in clinic yet. It has not been approved by FDA in clinic, but there are a lot of trials are going on to use these in a bigger way or in more cancer types. And it is doable if we can identify an antigen which will have a bigger impact. What are the toxicity which are associated with it? Definitely, these availability of the surface target. This is number one limitation in this area. If, that the unique targets, it has to be unique target, and if they can find more unique targets, probably it is possible to make it a broader. Severe toxicity, and I can tell you that almost half of the patients who are taking these CAR T cell infusion, they develop severe toxicity, and many of times it's fatal, but if they survive, they have a very good response rate. And outrageous cost, so if for one treatment, basically you are looking into at least $500,000, for one treatment. So you can think about the cost that, it is not covered by insurance companies, and it is like if you survive, you pay. If you die, you don't have to pay. That's how they can bring the patient, to the table, because these patients have failed all therapies. So for these patients, there is no hope. They are not getting any other treatment. So companies recruit them to these trials, and with the consent that there is a almost 50% fatality, with this, which is associated with toxicity. But if you survive, only then you have to pay. If you don't survive, you don't pay. Bad. But that's the reality. Here are some of the examples of the toxicity which we are talking about. They are often associated with the neurotoxicity or hemodynamic instability, which is, and one of the most common, side effect in all immunotherapies we have seen is something called capillary leak syndrome. You can see that, in our peripheral tissue, especially in lung, there are a lot of infiltration of immune cells gets into the lung because of this vascular leak syndrome, because it affects the endothelial cell permeability, and of course organ dysfunction. Altogether, in most of those fatalities are associated with multi-organ failure, which is a very common phenomenon if you have a massive inflammatory response. And the inflammatory cytokines, which are really major problem in this, will be your IL-6 and interferon gamma, and to some extent TNF alpha. These are the three cytokines which have been involved in most of the toxicity which have been associated in this CAR T cell therapy. And they have created a new term in this. They call it cytokine release syndrome. Okay. So what's the difference between syndrome and a disease? That's a general question. And if you don't know, probably we can discuss during the break, okay? We are not going to discuss right now. But think about it. What is the difference between syndrome and a disease, okay? In this, they call it cytokine release syndrome. So this cytokine release syndrome is not just restricted to CAR T cells. It happens in all other therapeutic protocol, whether they are using antibodies which are directly neutralizing tumor cells, or immune checkpoint inhibitors, are those antibodies which have been tagged toxins. You may not be able to read all these fine prints here, but general you have to remember is that most of the immunotherapeutic protocols which we have, it creates a massive inflammatory response, and that's a good sign actually, because that's the first sign that immune response is going to happen. But what happens is that these are uncontrolled inflammatory response, and that's what that cytokine release term is all about. And it leads to toxicity and then multi-organ failure. And again, the cytokines and which are involved in this whole process, of course, are different chemokines which are important in recruiting those inflammatory cells to various organs, molecules which are involved in vascular leak syndrome. But the molecules which have been really targeted in this are IL-6. There are a lot of IL-6 antibodies which have been used to treat these release syndrome, and also IL-17 which have been used to do that. TNF alpha antibodies are very commonly used because it is in the market for last 20 years and it is well tolerated, so they do use that also. So there are a lot of ways they are controlling the toxicity by dampening the inflammatory response. So this is not a good sign because you need to have a good inflammatory response to have the effective immune response. But you don't want these patients to die because of the inflammatory responses. So there are a lot of balance and fine-tuning which goes on in this. Here is a simpler cartoon about this whole cytokine release syndrome. Usually stimulation is because of the cells stimulation, activation, production of, and lysis of a lot of tumor cells, which can cause more and more inflammatory milieu. And the cytokines commonly, I will say TNF alpha, interferon gamma, and if you combine them together, it is a very potent inflammatory group. IL-6 is another one. And there is a whole gradation of symptoms which you can see depending on the patient. If it is grade 1 and grade 2, they can manage it with other inflammatory inhibitors like COX-2 inhibitors and so on. But as soon as it gets into grade 3 and grade 4, they may have to stop those treatments further, and they have to manage these toxicity first before they can go and treat these patients further. So there are different strategies to overcome these challenges in CAR T cell therapy. For cytokine release syndrome, they have anti-inflammatory approaches like blocking IL-6, using these pan-JAK inhibitors, which is commonly used in rheumatoid arthritis, or giving lots of steroids. Development of anti-CAR idiotypic antibodies against, especially with the murine SCFP, which are not commonly used in the humans. So basically, they are using now humanized SCFB in their recombinant technology, and hopefully the outcome should be the longer and persistence CAR T cell, because they don't want those CAR T cells to be cleared. Lack of persistence of CAR T cells, again, in normal immune response, usually these T cells, once they are activated and job is done, you want these T cells to die and be removed, because you don't want them to go and start attacking other cells. But in case of cancer, you want these CAR T cells to hang around longer, and in order to do that, there have to be a right approach and right kind of signal which have to be given to them to enhance their survivability. Relapse owing to the loss of CD19 epitope, like many tumors, as you are giving them these CAR T cells which are going to target all CD19 positive B cells, but there are some B cells which are not dependent on CD19. Maybe they are expressing some other markers like CD22 or CD52. So combining them with other CAR T cells probably will be the next step, which is a combinatorial surface targeting to prevent that kind of escape. So this is another simple generalization, which basically mechanisms to rescue CAR T cells is by activating them again, after the infusion and after the initial assault is done. You can activate them, using different cytokines, and you can prevent their exhaustion also, because as I said, these T cells, in normal physiology, these activated T cells to have either die or go under senescence. And that senescence or aging phenomena is often associated with these PD-1 and PD-L1 upregulation, which can be targeted by using these antibodies to PD-1 and PD-L1, which we will discuss later, and can reactivate these CAR T cells to continue to survey, and provide immunity against these tumors for a long, long time. Future applications, unlimited. Basically, here, if you can identify a nice, good surface marker, you can create it. It's all, engineering. You are engineering, something which, and we are pretty good at it. Ongoing advances in T cell engineering, gene editing, and manufacturing have a lot of potential. So there is a lot of hope in these T cell based therapies. It can be translated to other cell type, such as induced pluripotent stem cell, hematopoietic stem cell, NK cells, or other cell type, because we know how these cells work. NK cell is the best example. You know how NK cell kill the tumor cells. So you can basically, rather than taking T cells and doing what they are doing, they can do the same thing with the NK cell. Problem is very difficult to grow NK cell and expand NK cell. But for other cell types like stem cells, it is possible. Stem cells can be grown and can be expanded and can be utilized. So there are a lot of hope in this. It's not only for cancer but many other diseases. So that's what I had for CAR T cells. And if you have any question, we will continue to have that discussion during our break. So let's have some discussion on this immune checkpoint blockade. This is another hot area in cancer immunotherapy, which is generating a lot of revenue and a lot of hope. So the first one which have been targeted is CTLA-4. I think by this time you should know everything about CTLA-4. It is immunosuppressive, and immunosuppressive. So if you can block CTLA-4, probably you can enhance the response and antigen presentation, and outcome will be pretty good. So that's what is written all here. Okay, I'm not going to go and read it, but all we are trying to say here is that CTLA-4 is a very good target to do immune checkpoint blockade. The other one is PD-1, PD-L1. We didn't discuss more about it. PD-1 was first discovered as a member of immunoglobulin gene superfamily, thought to be involved in programmed cell death. That's how the name PD-1 came. Further studies have shown that this is very much involved in T cell function, not by death, but by old age. Okay? Keep in mind, that it's some sort of like a cellular senescence. That the thing which I was talking about is that once the T cell activation happens, through engagement of your signal 1 and signal 2, once the T cell are activated, you remember the expression of CTLA-4 goes up just to prevent further activation. And once that is happened, and once the function of T cell is done, then these T cells have to be either eliminated or put somewhere in on shelf in dormancy. And that could be, it's not a senescence in ideal way because they have not found the specific senescence marker. They have called it old age, or vidrashram. But these T cells basically are not able to kill, other cell types unless they have been reactivated, and that involves this activation of PD-1 and PD-L1 on the other tissue. When engaged, it mediates a complex epigenetic and transcriptional transformation in T cell, creating a new hyporesponsive phenotype called exhaustion that resembles like senescence. Okay? And if you block this in engagement, probably you can reinvigorate these cells to start doing exactly what they were doing before. PD-1 is triggered by B7 homologs like PD-L1 and PD-L2 that constitutively reside on non-lymphoid tissue and can be upregulated by immune cells in pro-inflammatory cytokines like interferon alpha and TNF. Many tumors actually express these PD-L1 and PD-L2. Not every cell within the tumor, but you can find a very pattern-wise, very heterogeneous expression of PD-1 and PD-L1. And supposed to be an important blockade to those T cells which are having PD-1, and then these basically will be non-functional. Interferon gamma and TNF again is a very interesting molecule here because in this therapy where they are trying to target PD-L1, PD-L1 antibody will only work if the cells are expressing PD-L1 and PD-L2. And to upregulate PD-L1 and PD-L2, they treat those patients with interferon gamma and TNF so that they can upregulate that expression of PD-L1, and then they can use those antibodies to block PD-L1 and PD-L2. So here is a simpler cartoon, blockade of CTLA-4 or PD-1 for anti-tumor response. In lymph node, you have T cell receptor antigen complex with MHC as a signal 1. And your signal 2 is your B7 interacting with CD28. CTLA-4 is a suppressor. So if you use antibody to CTLA-4, like this is one is ipilimumab. And this was the first immune checkpoint blockade antibody which was approved. Then you can basically enhance this whole response. Inside the tumor, tumor cells express this called PD-L1, and PD-1 is expressed on the T cells. Again, these cells which are expressing the PD-1, if you can block using anti-PD-1, or if you can block PD-L1, you can enhance the T cell responses. So that's a basic simple idea about this whole PD-1, PD-L1 therapy. In this, you can have these all happening at different stages, and that basically the basis for combining other immune checkpoint inhibitors are two of them together. Very early on, we all know and we learned that antigen presenting cells and T cells, how they interact and how the antigen is presented. And in this context, your T cells is basically activated, and then you have expression of the CTLA-4. And this CTLA-4 is important in keeping the immune response in check, but at least in cancer, this CTLA-4 is constitutively expressed on those T cells, because most often your T cells in cancer patients are always hyperactivated. But if we can block this CTLA-4, probably we can enhance overall the T cell response. But at that stage, they also have the expression of PD-1 and PD-L1. And combining these two together, probably will have a better response. But here, what they are saying is that later on, the CTLA-4 becomes the important regulatory molecule, and in a well-established tumor, the blockade of CTLA-4 is an important mechanism, an important way to enhance overall T cell responses. So here is the timing of clinical development of different immune checkpoint blockade. Most of them are in antibody. CTLA-4 was first developed in June of 2000. This is the first one which was approved. And you can see in the list right now, you have multiple of these antibodies which have been approved. How can you say that these are antibodies? When you look at the name, okay, and if you see this called MAB, what it stands for is a monoclonal antibody, okay. So that's one easy way. And if it says NIB, okay, it means these are small molecule antagonists or inhibitors against something, okay. NIB, okay, MAB and NIB. So most of these are monoclonal antibodies. They have been used either alone or in combination to treat different malignancies. And these are basically where they have been approved by FDA. But what happens is that once it is approved for one cancer type, now it's a clinician's decision to use it for other cancer type. They can use it, okay, because they have already gone through all the cytotoxicity and clinical experimentation. So it is easily, it is easy to combine multiple of these together. This was a study which was started in 2010, combining these CTLA-4 and PD-1 targeting in melanoma. The result of this study was published last month in New England Journal of Medicine, and I have one slide to show you. By combining these two together, basically you will have a synergistic response. It takes months to years to get this kind of result. It all depends on the tumor type. In some cases, you may have result within 2 years. In some cases, if the survival is somewhere 5 to 7 years, you are looking at the longer time period. PD-1 targeting, again, you are looking at the same picture again and again. Basically here, those cells which are expressing PD-1 are not basically functional. They get exhausted. So if you reactivate by blocking PD-1 or PD-L1, you can enhance these T cells, expand them, and probably they will be able to eliminate the tumor cell, enhance tumor cell killing, and so on. They have used this technology a lot now, so there is a very well-defined mechanism, but there is not so much known what goes downstream in this box in T cells or in cancer cells. But it is very clear that regulation of PD-L1 expression, PD-1 expression will be some area where there will be more opportunities to find ways to make cancer cells more susceptible to these kind of treatments. And this is a simpler way, I think in the beginning itself we were talking about some tumor cells are easier to treat using different immunotherapies. And those tumors usually often have a lot of infiltration of different immune cells where it is easier. If you activate them appropriately, probably you will have a better response. For some cells, for some tumor types, they consider or they classify them as a non-immunogenic. They call it hot tumor or they call it cold tumor. And in cold tumor, most often you have a very few infiltrates, and the recruitment of those cells are limited. So you can make those tumors immunogenic first, and then you can do the immunotherapies, and probably you can have a better durable clinical outcome. You can see the same thing, not just depending on the patient population. Different patient will have a different type of tumor within the same tumor type. But even within the cancer patient, you may find that some of the foci, because we are talking about the micromets here now, and some of them will be more susceptible for immunotherapeutic treatment, and some of them will be not. And that heterogeneity basically is going to be a major challenge in terms of response to these immune checkpoint blockade. They were approved in 2000. Now many of them are actually approved in 2010 and later. So it's almost 10 years they have now experience. And every time you have a novel therapy comes into picture for cancer, it is always followed by resistance. So the resistance, those tumors which are resistant to this kind of immune checkpoint inhibitors, are these kind of tumors where either there are no infiltrates, lymphocyte infiltrates, or those tumors, are those individuals who had micromets which were non-responsive to these treatments. So there are other approaches which people have tried to use it to restore or make the tumors more hot, and this actually was published again last month from the same group, Jim Allison group. Here Jim Allison is the Nobel Prize winner. He got Nobel Prize last year, and his wife Padmini Sharma. So this is husband-wife team. She didn't get Nobel Prize, by the way. He got the Nobel Prize. There were two other recipients. But they still have a lab together, and this is a work which they published this is in bone metastasis for prostate cancer. And again, when it comes to immunosuppressive cytokine or growth factor, the most important one is TGF-beta, okay? TGF-beta is a highly immunosuppressive, and it is important in bone metastasis. If you were there in my talk when I was giving in NCCS, TGF-beta was one molecule which was coming out, to support, in establishment of metastasis. So in this case, basically, if they use just, they are comparing the soft tissue versus bone environment. In soft tissue, this is just looking at the tumor into two different environments. So in soft tissue, they had a very nice response. These immune checkpoint blockade inhibitors was successful in removing the primary tumor. This is in prostate. But when you look at the bone metastasis in these, this is done in animals. So in these animals, basically, bone metastasis was not affected. And this was because of the production of TGF-beta, which suppressed that response to. But if you combine or if you block the TGF-beta activity by using anti-TGF-beta antibody, which is actually now clinically available, it is approved for clinical use, then you can potentiate the response to immune checkpoint blocker. So sometimes maybe you will have to go and do more combinations to get, depending on the environment, depending on the immunosuppressive environment within that tumor. And this is the best example to look at the site-specific responses for these immune checkpoint blockade. The adverse events which are often associated with immune checkpoint blockade are again what you see with most of the immunotherapeutic protocols, which is inflammatory side effects or autoimmunity. Because those antigens which are targeted in this case also express on the normal cells. So the normal organ, or any organ which expresses that antigen, can also be affected. Those T cells are not just limited to the cancer itself. So there is a global tempering of immune homeostasis which goes on in these patients who are undergoing these treatments. And often, if you look at the overall success, not just like every other immunotherapies, the cost of breaking tolerance is that checkpoint blockade treatment is accompanied by immune-related adverse event in almost 80% of the patient. So there are a lot of management which goes on in these patients who are undergoing these kind of treatments. Again, overall, if you look at, these patients are not going to get cure. There are a lot of optimism in these treatment plans. The best success they have is trying to delay death. So basically, it is increasing the overall survival. And that survival hope is that, if it is even, because since these kind of treatments, or at least these treatments, were applied to those individuals who have failed everything, and if you can prolong their survival even for 3 months, hope is that, you are making these cancers more susceptible to these kind of treatment, and maybe other immunotherapies together with this will be a better will have a better outcome. So there is a lot of opportunity for multidisciplinary collaborations, or combination treatments or therapies to make it more and more effective. So there are combination therapies which have been planned very early on. As I said, the trial started sometimes in 2000, and one of the best success is combining the CTLA-4 to PD-1, which is very successful. And there is a biology behind it, because what CTLA-4 does and what PD-1 does. Probably the outcome should be very different. And if you look at all different kinds of combination therapies which are going on along with these immune checkpoint inhibitors, and list is pretty long. Actually, there are a lot of clinical trials going on. Right now, I think the last number which we had in our ACR meeting last year was that there are 3,600 clinical trials going on worldwide to use these clinical immune checkpoint blockade with the different kind of therapies, targeted therapies, chemotherapy, radiation therapy, chemoradio, or multi-way combo, or other therapies. And in most cases, you can see chemotherapy is a big picture here, radiotherapy or chemoradiotherapy. But there are a lot of trials which there are more than 300 trials going on combining it with the CTLA-4. Other small molecule inhibitors and other growth factor receptor targeting has also been combined together. But all in all, there are a lot of combination strategies which are going on because the basic understanding is that no matter how successful this immune checkpoint blockade will be, it is not going to be a monotherapy. So this is a success story which I was talking about. Here they are looking at the median treatment-free survival. Means these patients, they got the treatment, okay, and they went home, and they don't have to come back to get the second treatment, okay. And this is in malignant melanoma. You are looking into months, and if they just get CTLA-4 antibody, you are looking at a couple of months, of treatment-free survival. in treatment-free interval, not survival. If you have PD-1, you are looking into the same number, somewhere like 2 months. And these patients were, given both of them together, you are looking into 20 months, which is very unexpected in case of melanoma. These are metastatic melanoma patients who have a very high mortality. Within a year, most of them they die, and there is no other treatment which is available. So this kind of data is something which is very, very encouraging. And they all know that these, although they are very expensive, but for these patients going from 2 months to 20 months, you don't need any statistics. Okay, this is a very significant information. It just came out after last 7 years of clinical trials. Just published this. So what I can say is that not all cancer types can be targeted by using checkpoint blockade, and this can be used in many other ways in combining with other therapies, probably to make the outcome better. While basic science research has changed the face of clinical oncology, efforts must continue to uncover molecular pathways that drive immune function and homeostasis. I told you that everything what we know today is everything on the surface. Okay, every target which have been used in immunotherapies, it's all on the surface. What happens downstream is a mystery. Okay, and I don't know how many of you know the work of Dr. Saha. I can give his example. Okay, how many of you know his work? What he does? Have you heard him giving lecture here? If not, maybe you should invite him, okay? Dr. Saha. He works on 4BB, okay, 41BB, okay. This is one of those co-stimulatory molecule, you know, CD40 ligand. He works on one of the CD40 ligand. CD40 ligand is one of the molecule which is important for enhancing antigen, you know, presentation. Engagement of that is very important, but it all depends on the dynamics, how much CD40 ligand. It can be anti-inflammatory, it can be pro-inflammatory, and his whole thing is downstream. Why, you know, depending on the, you know, these anti and pro-inflammatory milieu, just with one receptor targeting, why it is so different. An immune response, it is like that, okay? The same receptor can be exploited to have those two different kind of response, and it's all happening downstream. So immunology, so far whatever we know, even whatever I showed you and discussed, I didn't talk about any downstream molecule other than zeta chain, okay? You know, so there is nothing, you know, no kinase, nothing. But there are a lot of things going on right underneath, okay? And that's an open box for future scientists to explore. Probably you will find something maybe which will be common for all of these problems which we are talking about, you know. And this is one thing which will be driver, you know, to have all these different phenotypes which we are talking about. And if you can find that driver, probably you can have a better outcome. And it is possible. It's not a dream. Although the baton of immunotherapy has been passed to the clinic, the history of this field has taught us that strong basic science effort in the laboratory today are crucial to produce innovative innovation at the bedside tomorrow. What happens is that all this hype we have, you know, evening news, you know, all the advertisement, and everybody is very hopeful that this is going to be a cure. It comes with a disappointment also, you know, because the success rate which we are talking about is 20%, and if any, you know, any adverse event happens, and if there is any, you know, headline news comes out of this, which is bad news for this, it will shut down the whole thing, which is not what you know, this field needs. So what it is basically trying to tell is that clinicians are always hyper, you know, they always, you know, take things very differently. But as a basic researcher, you know, we all have to be careful and always try to understand it better and better, probably because, you know, that everything lies, you know, in our understanding and increasing our knowledge. We cannot do everything by ourselves, and that's where the community comes into the picture. So always collaborate, try to learn, try to study, read a lot, you know, because reading always makes you a little better, and you get more information. Just don't read on your cell phone, you know, try to go to library or at least print out something someday. Okay, go beyond the title, you know, look into the abstract or somewhere else. Okay. So all I'm trying to do is that, you know, there are a lot of opportunities to explore more and more new novel things in this area, and this area is still very young in you know, as far as I am concerned. Thank you very much.